Hello dear students, welcome to our biology program. Today we are going to complete immunity in men and especially the component of the lymphatic system. Let's begin. Last time we studied the main components of the lymphatic system, but today we are going to complete the last point of it, which is antibodies. Then what is an antibody? An antibody, it is a specific protein called immunoglobulin, which is symbolized by Ig, and it has a very important shape, which is Y shape. All the antibodies are present in the blood and other fluid of the human and other vertebrates. Then from where they come? They come from, or they are produced by antibodies secreting plasma B cells. Very important function. Antibodies and the complements adhere with bacteria cells and other foreign molecules to offer them to other white blood cells to engulf them. Let's look to this film. Look here, the bacteria enter the body, and you found that there is antibodies with their very recognized shape, the Y shape. They adhere this bacteria or any foreign body, and they introduce it to another white blood cells, which is the to engulf it as a macrophage, for example. These antibodies, how they are formed? The surface of the bacteria cells and the foreign bodies that invade the body tissue have component called antigen. On the surface of B lymphocyte, there are receptors. When we looked to this film, you found that there is a main points on this bacteria where the antibody bind to it. And these receptors recognize and join with antigen on the surface of bacteria cells or foreign bodies. Then the B cells change to specialized B cells called plasma B cells. Then what is the importance of these plasma B cells? These B cells, plasma B cells, in turn produce antibodies. When the B lymphocyte join with antigen for the first time, they divide many times to produce groups of cells. Each group produces a specific type of antibody against a specific type of antigen. So this antibody, it is a specialized one. So each type of antigen has its own type of antibodies. Then how many types of antibodies we have? We have 5 types. So these types are the IgA. Remember that the symbol Ig, it represents the immunoglobulin, which represents the antibody. So we have antibody A, antibody E or IgE, IgD, IgM, and IgG. These all 5 types, each one of them, it has its own antigen which bend to it or recognize it. Then if we knew the types and how this is formed, how this antibody is formed, and we knew then what is the structure of this antibody? You are going to see that each antibody is consisting of 2 main chains. The 2 chains, each one of them is composed of a polypeptide chains. 2 of these chains are long and they are called heavy chains, and the other 2, they are of polypeptide chains, are called light chains, and they are short. These 4 chains are joined together by a disulfide bond. And then this disulfide bond and all these 4 chains have a nest, what we call it a nest or a base, on which all of this is formed, and this is called the constant part or the constant region. Then let's look another time for these chains. The polypeptide chains consist of 2 sites. The 4 chain, as I told you before, they are joined together by a disulfide bonds. Each polypeptide chain consists of 2 sites, or the long polypeptide chains consist of 2 sites. The antigen binding site and variable site. Each antibody has 2 identical antigen binding site. Antigen binding site. The shape of these sites is different from one antibody to another because of the variation of the amino acids. So these antigen binding sites, they have different shape because of the variation of the amino acids which binds to them, forming the polypeptide bond according to the amino acid sequence, type and spatial shape which specify the antigen, which specify the antigen. So this amino acid make the shape different of this binding site because of the variation of the amino acids, and this amino acids has its own specified antigen or its own specific antigen. These sites help the binding between an antigen and its specific antibody, and which resemble the lock and key, because it matches the variable site of this specific antibody like mirror image of a specific antigen. This binding form an antigen-antibody complex. So once they are joined together or binding together, it forms what we call it antigen-antibody complex. And the last point or the last part of this structure, it is the constant region, which is the base or the nest of this antibody. Constant region, or which is symbolized by the symbol C or the letter C, is the nest of the antibody because it has a constant shape and structure in all types of antibodies. How these antibodies work, or what are the mechanisms of antibodies? Ways of antibodies mechanism to stop the action of antigen. As in the figure, we found that they are 5 types: neutralization, agglutination or clumping, precipitation, lysis, and antitoxins. We are going to study each one by one. Antibodies have 2 antigen binding sites only, while antigen have many binding sites, which make a confirmative binding certain between the antibodies of their antigen and the other one. Neutralization. As you see from the most important function of the antibodies and of the antibodies in resisting virus, in neutralizing this virus and stopping their activity through. Prevent the virus from adhering to the membranes of the host cell and from spreading or passing inside them. Prevent the nucleic acid of the virus from replicate. If the virus succeeded in penetrating inside the host cell's membrane, the antibodies will prevent the nucleic acids of the virus from coming out of their protein coat and replicate inside the host cell by keeping the coat intact or sealed. So they prevent the nucleic acid of this virus from duplicated inside. Also, it prevents the virus from adhering to or coming near to the membrane of the host. The second type is agglutination. Some antibodies has many antigen binding sites, such as IgM, which make them able to bind to more than one antigen. This leads to the clumping of microbes on the same antibodies. This makes them weaker and liable to be engulfed by the phagocyte. Look here, you found that there is some bacteria here and they are engulfed by it because of their agglutination, or they coming together, they are banded together by the effect of this antibody. Now, the third mechanism is precipitation. This occurs in the soluble antigens, in which the binding between antibodies and these antigens leads to the formation of insoluble antigen-antibody complexes, which form a precipitate to facilitate its engulfing by phagocytes. The fourth mechanism, it will be lysis. This takes place by binding between antibodies and antigens activates a specific protein and enzymes called complement to lyse the coat of antigens and dissolve their contents, which makes them easily engulfed by phagocytes. The last point, it will be antitoxins. In antitoxins, we found that the antibody bind to the toxins which are secreted from the foreign bodies or the bacteria, virus, and so on, and form a complex compound. So the antibody bind to toxins and form complex compound of antibodies and toxins. These complexes activate the complements to react with them in a chain reaction that leads finally at the end to detoxifying them. That means that these toxins will be useless, or there is no effect for them, by changing them into detoxifying them, and also make them ready to be engulfed by phagocytes. Now we knew the mechanism of these antibodies, and this was the last point of the components of the lymphatic system. We knew the components of the lymphatic systems. We knew from these components the lymphocytes, the white blood cells, macrophage, assisting chemicals, and many things also. And finally, the antibodies. And after that, what is the immunity, or this immunity system in man, and how it works? Immune system mechanism in man. The immune system in man works through two systems. The first one, which we born with it, or what we call it naturally, or natural immunity, or another name which is called non-specific or innate immunity. Non-specific has no specific antigen it works on them, or a special pathogen it works on. And another type of immunity or system which is called acquired specific or adaptive immunity. So the acquired immunity and natural immunity. These two systems of immunity, we are going to deal with them today. First one, the natural, non-specific or innate immunity. But we have to say something before, that these two systems, although they are different, but they work together in a harmony, or they cooperative with each other. They activate, by this harmony, they activate each other to help the body to deal with pathogens successfully. Then let's go and study the first one. If you look for the body, you found that the body, it is covered from the outer side by a very, very wide cover, which we call it the skin. So, and then you look to the mouth, you found saliva. This is also facing the microbes directly. So this one, we call it the first line of defense. Then a little bit, once this enter to the inside of the body, we found that this, if it enter to reach the blood, at this moment we are in the second line. So the first line, it is of defense, it is the outer side of the body, or anything which face directly the microbes or the pathogens. The second line, like the inflammatory or the inflammatory responses by injured and so on, we found some pus comes out from it. This because of the battle which is between the white blood cells and pathogen. Then let's begin and study the natural non-specific immune system. First of all, it is a group of defense mechanisms that protect the body and is characterized by rapid effective response to resist, fight, and destroy any microbe or foreign body that invade the body. These mechanisms are non-specific against certain microbe or antigen. These mechanisms are classified into three lines of defense. The first line, the main function of this line is preventing pathogen from entering the body. It includes a group of physical or natural barriers in the body, such as the skin. The skin here, it is covered by a horny layer on its surface, which acts as a barrier that is too difficult to be penetrated or to pass through. Then there is another part, which is the sweat, which is secreted by the sweat glands on the skin surface. Kills most of the microbes because its salinity kills the microbes. There is another one, which is the cerumen. The cerumen, it is secreted by the ears on or what we call it ears wax. So the cerumen, it is the ears wax, and it is secreted by the ears to protect it from microbes by killing the microbes. First line defense, which is called the tears. The tears here, it contains antimicrobial substance, and these tears protect the eyes from the microbes by killing them. And once we glance, so we clean the eye from this by covering it with these tears. Another part, which is the mucus in the respiratory tract. It is a viscous fluid that lines the bronchi to adhere with the microbes. Mucus traps microbes and foreign bodies entering with the air. Then the mucus, together with the trapped microbes, are expelled to the outside of the body by the action of the beating cilia lining these tracts. Another one, which is the saliva, contains a substance that kill microbes in addition to enzymes that can dissolve microbes. The acidity of the gastric juice. Look to the film here. You found that the nose enter from the nose the air which contains some pathogens and foreign object, and by the effect of this cilia and the mucus they are expelled outside the body. And here the gastric juices, which is secreted by the epithelial lining of the stomach. Look here, they are here lining the stomach, produces and secretes the strong hydrochloric acid that can kill microbes entering with the food. The second line defense, this system acts in the pathogen succeeds in penetrating the first lines of defenses and invades body tissue through a slash in the skin. This line of defense is an internal mechanism in which the body uses successive non-specific mechanisms that surround the invading microbes within second or minute to prevent the microbes from spreading. This mechanism that start by a severe inflammation. Then what is inflammation? Is an immediate response of the body tissue injured by a foreign body like bacteria, in which some changes takes place in the area of injury. Inflammatory responses is the non-specific defense mechanism in the area of injury as a person to the damaged tissue caused by the injury or by infection with pathogen. The mechanism of this second line defense, when microbes or foreign body invade the injury body tissue, some changes takes place in the area of injury where specific cells such as mast cells, eosinophilous cells, and the T lymphocyte cells secreting large quantities of inflammatory generating substance like histamine, some interleukins, and other substances. The role of the inflammation generating substance dilate the blood vessels to maximum limit at the area of injury. The second role of them, increase the permeability of arterioles and the capillaries to blood fluid, which then leak from the blood circulation. This leads to the swelling of tissues in the site of injury. Also, it allows the passage of chemicals that kills and dissolves bacteria to the site. Also, they increase in the permeability of blood vessels, also enable white blood cells as neutrophils, monocytes, and macrophage to fight and kill foreign bodies and microbes. First question. Write the scientific term for each of the following statements. A type of cells which store information about antigen fought or killed by the immune system in the past. Yes, it is the memory cells. Another scientific term, the response of immune system to the infection of the same disease again. Secondary immune response. Very good. The resistance of the body to pathogens that infected the body before. Acquired immunity. The immune response done by T lymphocyte, which is done by the receptor found on its membrane that give it a specific response to antigen. Cellular or cell-mediated immunity. A series of specific defense mechanisms that the lymphocyte perform to resist the pathogen. It is the immune response. Then what about the comparisons? Look for these two comparisons. Compare between acquired immunity and natural immunity, primary and second immunity response. Let's begin by the natural immunity and acquired immunity. The natural immunity, it is a group of defense mechanisms that protect the body, while the acquired immunity, the resistance of the body to pathogen that infected the body before. In the natural immunity, non-specific or innate immunity, while the acquired, they are specific or adaptive. Natural immunity do not make memory cells. The acquired immunity form memory cells. The first line of defense includes the skin, mucus, tears, sweat, and hydrochloric acid of the stomach, secreted by the stomach. The second line of defense, inflammatory response, represent the third line of defense, and it is subdivided to two mechanisms: humoral or antibody mediate immunity, cellular or cell mediate immunity. And now, my dear students, we reached the final point of our program. I hope you enjoyed it. See you next time.